Chapter 11, the first of two, where we'll be looking at how inheritance works. The other thing in this chapter is meiosis, which is how we make gametes, or eggs and sperm. So first thing you need to know, we mentioned this before, that chromosomes come in pairs. Those pairs are called homologous chromosomes. As I said in, I think it was chapter 7, the homologous chromosomes have the same genes on them. Organisms that have two copies of each chromosome are called diploid. That means basically two sets. We each got one of those chromosomes from each parent. And our total number is 46, with basically 23 different ones, although that 23rd pair, the X and Y, are a little different. So this illustrates, on the left we have the karyotype, a picture of all the chromosomes. And they're arranged in pairs, as we saw before. And then we've taken a close-up of chromosome 7, which has a particular gene that they're looking at here called CFTR. That's what we're going to be looking at as our example. Okay. So, setting aside the chromosomes for a minute, we're going to look at the cells and production of gametes, or sex cells, the cells that are used to make a new organism in sexual reproduction. They are called haploid, which means they have only one copy of each chromosome. And I know it sounds like it means they have half a set, but it really does mean one set. Meiosis is the process of dividing a cell so that you get half the DNA in it. What it does is it makes 4 daughter cells, each with half the DNA, instead of 2 with equal DNA as we have in mitosis. What it does is it copies the DNA, but then it divides twice. We call those two divisions meiosis 1, which produces 2 cells, and then meiosis 2, which produces 4 cells from dividing those first 2. Now, it's not exactly mitosis and then splitting up again with only one copy from each homologous pair. In order to make sure that all the cells get one from each homologous pair, they line up a little differently. So this shows just one pair of chromosomes, but if we had multiples shown, you would have the way we had our single X's in metaphase in mitosis. You would now have another X next to each one that's its homologous chromosome, instead of them just being in a single line. And then when the cell divides, in anaphase, it separates the chromosomes, one from each homologous pair. And so instead of getting the single chromosome, the daughter cells get the 2 chromatids together, but they only get half as many. So a human cell that has just completed meiosis 1 would have 23 chromosomes, but with 2 chromatids each. And then in mitosis 2, it's exactly like meiosis 2, it's exactly like mitosis, except you're starting with only half as many chromosomes. And so the cells at the end would have 23 chromosomes again, but only 1 chromatid each. So this illustrates the whole process. They've used colors to indicate the pairs. Now there's 2 things that happen in meiosis that help increase the variability in the gametes. So how many different gametes the same organism can produce. These are called recombination and independent assortment. Recombination is something that happens while those homologous pairs are lined up together during prophase 1. And what happens is they can get tangled together, and then when they separate, each one has a piece of the other. And so what you see here is 4 different chromatids. One that's all from the person going, one that's all from the grandmother, one that's all from the grandfather of whatever organism these would join to create. And then you have 2 that are partial of each. And so that results in you don't have to get the entire chromosome from one grandparent. You can get partials. So this chromosome that you get from your mom could be part grandma, part grandpa, or it could be all grandma or all grandpa. And if it's parts, which parts can vary as well. And so you get a lot of different possible chromosomes from one organism going through meiosis. Independent assortment is just the fact that each pair of homologous chromosomes can be separated in any direction. So they're not going to be guaranteed to line up with all of your mom's chromosomes on one side and all of your dad's on the other side, and then so you make one egg or sperm with only grandma's chromosomes and one with only grandpa's chromosomes. They can line up any possible way. You can end up with anything from all from one grandparent to 22 and 1 to 11 and 11, or 11 and 12. Okay. And so even if they didn't have recombination, you would get some genes from each grandparent in the gamete. And so then this illustrates by color, and they've used 2 different letters, well, the same letter, but a capital and a lowercase to indicate a particular gene on that chromosome. And then you have 2 different gametes based on one lineup, but if they line up another way, you get 2 more different gametes, and then a third way they could line up gets yet another set of 2. And with 23 pairs, there's a huge number of ways they could line up. This says about 8 million. Okay. Now, how we get those haploid gametes together is called fertilization. The cell they form is called a zygote. Zygote means yolk, not like an egg yolk, but like a yoke that you would use to put on 2 oxen or horses that are going to be pulling a plow together. So it's a joining. You get 1 set of chromosomes from the egg and 1 from the sperm, and so that makes a diploid zygote. Again, 2 copies of the genome, all homologous pairs of chromosomes. And then as the zygote grows and divides, it is called an embryo. In human development, after about 8 weeks, it's then called a fetus. So here's 1 egg and 1 sperm from meiosis. Here's the zygote, single cell. And then the early stage of the embryo, it hasn't started taking any shape yet, but it's made of multiple cells. And then eventually it develops into the whole organism. Right, moving now into genetics. So, what combination of alleles you get for any particular gene is called your genotype. You get 1 from each parent, so you have 2 genes, or 2 alleles in the genotype. The 2 possibilities are that you have 2 of the same allele, and that's called homozygous, the same pairing. Or you could have 2 different alleles, and that's called heterozygous, different pairing. The phenotype is then what trait you express. This is determined by the genotype. It affects what form of the protein you're going to make. If you are homozygous, then your phenotype matches whatever the allele says. If you are heterozygous, which gene, which allele are you going to express? That's going to vary depending on the gene. The way traits are passed is called modes of inheritance. The basic pattern, which applies to all diploid organisms, was discovered by a monk named Gregor Mendel in the 1860s. We're going to look at his work in the Milestones in Biology. He worked with pea plants, but they're also diploid, so the same principles apply. So the most common pattern, or the simplest pattern at least, for gene expression is called dominant and recessive. A dominant allele is one that is expressed even if you are heterozygous. So essentially, you only have to have one copy of that allele to have the trait it codes for. A recessive allele only shows if you are homozygous, that you don't have another allele that's dominant to cover it up. When we use abbreviations for genes, we use a capital letter for the dominant and a lowercase letter for the recessive. Now, on the lab, I put a capital D and a capital R, and that was just to indicate which trait was dominant, which was recessive. When we are looking at a gene, we're going to use the same letter always. Which one you use doesn't really matter, but you need to be consistent. So, how the inheritance works. With dominant alleles, you have the phenotype of that allele. You have to have gotten that allele from at least one of your parents. So most likely, that parent has that same phenotype. It's also possible that you have a new mutation, but that's not very common. What makes an allele dominant is that you need only one copy of it to make enough of the protein to see its effects. Often this is because the dominant protein is functional or does something visible, while the recessive protein either doesn't work, but it's okay to have only half as much being made and it's still enough, or that its expression is not something detectable, unless it's the only one there. Recessive alleles can be inherited without either parent having the phenotype. That would mean that the parents are heterozygous. That's also called a carrier. We call it that because they are carrying the recessive allele, even though they have a dominant phenotype. So when you talk about something skipping a generation, say grandma had blonde hair, both your parents have dark hair, and then you have blonde hair, that's because blonde hair is recessive. It's not something that's always that predictable. It's not always going to alternate generations. So some examples of dominant and recessive traits. So any kind of darker pigment like brown hair or brown eyes is usually dominant. A disorder called Huntington's disease is dominant. This is a disorder that starts to develop symptoms around age 40 or 50, and you would get the disorder even if you have only one allele for it. And then there's another disorder called polycystic kidney disease that can be dominant. There are multiple types. Okay. And so in this example here, we're showing the brown hair as the dominant. These two brown-haired parents have produced 2 blonde kids and 1 brown-haired kid. And I just got to tell you, this one I would have in class. My high school biology teacher had brown hair, and so did her husband, and their son had blonde hair. And when people would go, oh, we know how that happened. Of course, she's a biology teacher. She knows how it really happened. But you know what people are implying. The husband would say, yeah, I admit it, I was fooling around. Some examples of recessive traits. Most genetic disorders are recessive because the allele codes for a non-functional or less functional protein, and if you have one dominant allele that makes the functional protein, then you get enough of that protein to function properly. Cystic fibrosis is an example. We're going to start part 2 with that. Metabolic disorders such as PKU. This is an enzyme that doesn't work properly. And being colorblind is a recessive trait. If you have one functional allele for the receptor in the eye that picks up red or green, then you can distinguish between red and green. Now, colorblindness has another feature to it. It's what's called X-linked, meaning the gene is found on the X chromosome. That's going to be more complicated, so we'll come back to that in chapter 12. Essentially, it affects how sex affects how likely you are to have the recessive phenotype. The other thing we need to look at is probability of inheritance. Okay. First, I'm going to look at it from the single gamete from a single parent perspective. If the parent is heterozygous, they have 2 different alleles. Each offspring has a 50% chance of getting a particular allele. The other 50% is getting the other one. If the parent is homozygous, they only have one allele. They just have 2 copies of it. And so all of the offspring will get it. When we include both parents, this is when it gets more complicated. And so we have some tricks for illustrating the possibilities. The major one is called a Punnett square. You take the possible gametes from one parent and put them across the top, the possible gametes from the other parent and put them on the side. In this example, both parents are heterozygous, so the capital B is dominant, the lowercase b is recessive. And then the boxes indicate what genotype you would get if you combined the alleles from the side and from the top. So with 2 heterozygous parents, you have 1 chance of the child getting the dominant allele from both parents, 1 chance of them getting the recessive allele from both parents, and then the other 2, they've gotten 1 from one parent, 1 from the other parent. And so because there are 4 possibilities, our probabilities are in 25% blocks. Now, some rules for doing a Punnett square. Remember this when you work on the first genetics worksheet especially, but you will still need it for the second one. For any given gene, you choose a letter and stick with it. Okay. So we wouldn't put capital B, lowercase f. It's all B, or all D, or all F, whichever letter you decide to use. If there are more than 2 alleles that are dominant, or more than 2 that are recessive, then you'd use this little superscript to indicate it. Okay, so what we've got in this example is both parents are heterozygous, but they have a different dominant allele. And so we keep that superscript as we go through and match up the gametes. Okay. So first rule, use 1 letter. Second rule, if there's a dominant gene, you always list it first. And so in each of these parental boxes, we've put the dominant first, the recessive second, and then in the offspring boxes, even though they're getting them in a different order, it's dominant first, recessive second. So here's an example of both heterozygous parents, same as I've had before. And then in this example, we have one parent is homozygous recessive, the other is heterozygous. They've also marked the dominant allele with red, so you can see easily the chances of children getting it.